Hello everyone, welcome. In this video we're checking out variant 2 of May-June 2023. Before starting, make sure to check out the free notes I have available for you in the comment section down below. Now let's go ahead and start with question number 1. The sinoatrial node, SAN, and atrioventricular node, AVN, have an important role in the control of the cardiac cycle. The timing of the atrial and ventricular systole and diastole must be controlled so the blood passes through the heart efficiently. He says here, figure 1.1 is a summary of blood flow through the right side of the heart during one cardiac cycle, and 3 boxes on figure 1.1 are not complete. So blood arrives from the superior and inferior vena cava, blood then passes into the right atrium, then there will be atrial systole at this point, so that blood contracts out of the atria contracts so that blood flows into the ventricle, and at this point we'll have ventricular diastole. You will not have 2 systoles at once ever. Blood is forced into the right ventricle, then the atria are going to relax, so that's diastole, and then the ventricles are going to... Now, when the blood is forced, actually, we're still going to be at diastole for the ventricle. After that, blood leaves the right ventricle and enters the pulmonary arteries. Here, the ventricles contract, so systole, but here the atria relax and it's diastole. And so this is it. This brings me on to B. Impulses sent out by SAN pass to the AVN where there is a short delay. Now, with reference to figure 1.1, explain why it's important for the cardiac cycle that there is a short delay at the AVN after the impulse has been sent out by the SAN. So at this point over here, there will be a delay. You see, it doesn't immediately, contract. So here, as soon as blood enters, you see what happens is that the impulse from SAN, it goes to the atrioventricular node. Now, first, when the impulse from SAN, it goes through the atrial walls, and so it causes the systole of the atria. But after that, the impulse is going to go to the atrioventricular node. Now, at the atrioventricular node, the impulse is delayed so that we have a point where both of them are relaxing, so that blood is not contracting, so the atria and ventricles do not contract together at the same time, because that's dangerous for the heart. So that's the reason why. Okay, the reason why there's a delay is because to avoid contraction, or let's say systole of atria and ventricles at the same time, so that there's time so that ventricles, or let's say the blood first, so that the blood has time to leave atria and fill out ventricles. And I think that's important so that the ventricle acts as an efficient pump. Now this brings me on to C. Changes in blood pressure occur in the heart during the cardiac cycle. These changes cause the opening and closing of the bicuspid and tricuspid atrioventricular valves and the aortic pulmonary semilunar valve. So what happens here is changes in blood pressure cause the opening and closure. Explain how the blood pressure changes causing the opening of the tricuspid and the opening of the pulmonary, and help the blood flow. Yes, so remember, there is the tricuspid valve, and then there's also the pulmonary valve or the semilunar valve, and then help the blood flow through the heart. Okay, so it's very simple. Okay, blood only flows when there's blood opening of the tricuspid valve happens when there's a high pressure in the atria, low pressure in the ventricles, because here they're asking us to compare the blood pressures in the heart. And so yes, there's a high pressure in the atria, low pressure in the ventricles, causing the opening of the tricuspid valves, and then semilunar valves or pulmonary valve is going to open when there's a high pressure in the right ventricle, and obviously a low pressure in the arteries and low pressure in the atria in general because they're in diastole. So that's the reason. Okay. And there is an increase let's say right atrium during systole or atrial systole, and a decrease in blood pressure in ventricles. Obviously here it's ventricular diastole. Then, yes, that's the opening of this guy. Now, whereas for the pulmonary valve, it opens when there's an increase in blood pressure in the right ventricle and a decrease and a decrease in the right atrium. Obviously here define this as ventricular systole. Also, low blood pressure in pulmonary artery. Okay, now this moves me on to 2.1 is a photomicrograph in a longitudinal section through a root tip. Two different regions are visible, the root apical meristem and the root cap. Remember, the meristem is a point where lots of division is going to happen, so it's the growing part of the root. So you have the root apical meristem cells, and here we have the root cap cells. And he's saying cells in the root cap synthesize a gel-like sticky secretion known as a mucilage, which is important in reducing friction between the soil and the growing root, and it's composed mainly of polysaccharides, and it contains some amino acids and enzymes. So just to reduce friction between soil and growing root, we have mucilage, which is made just like mucus. It's made of polysaccharides and some amino acids, as well as also it contains enzymes. Interesting. So describe three differences visible between the root apical meristem cells and the root cap cells. So here, make sure to refer back to the photo and let's compare. Now, as you can see here, we can clearly, obviously, like by our logic, we know that the root apical meristem cells and the ones that they're pointing out to very specifically are in different stages of the mitotic cell cycle. So you can see some of them are in interphase, some of them are in prophase, some of them are in metaphase, and here we have, I think this is anaphase because they're pulling apart. Okay, what else we have here, for example, anaphase, as you can see in this one, metaphase in this one. So they're in different stages of the cell cycle, whereas all of them here are not going through mitosis. So basically here they are going through mitosis, but the cells in the apical root are actually, or root cap cells, they're not going through mitosis. So cells in root apical meristem like, for example, metaphase, anaphase. Whereas cells in root cap aren't going through mitosis. Now, for the second visible difference, we can see these cells here are larger in size, whereas the cells here are much smaller. And this also makes sense because cells here, they first, are formed, but then they move here and they grow, they gain volume because their vacuoles fill up with water. And so cells here are bigger, and also their nuclei are bigger. Here you can see visible nucleoli, like you can see this, but here their nuclei are just big, but they don't have any nuclei. What else can we see? You can see the cells here are, what's it called? Here the cells are more rectangular in shape, but here they're different. They're also rectangular, but like some of them are different in shape. However, I would not point that out. I'll just point out that they are bigger and they have bigger nuclei. And another difference would be the visible nucleoli, I suppose. What else could we say? I think that's what I can spot. Obviously, you can spot different things. Let's go ahead and write that down. Okay. So cells in root cap are bigger with bigger nuclei compared to cells in the apical meristem, in the root apical meristem. And then lastly, we can say cells in the root apical meristem obviously when an interphase. Okay, this moves me on to B. Mucilage acts as a glue to bind tiny soil particles together, forming small clumps close to the root. These small clumps help to maintain soil water around the root tip, preventing the loss of water. Okay, now with reference to the cohesive and adhesive properties of water, suggest and explain how the formation of small clumps of soil helps to maintain the soil water. Yes, around the root tip, so helps to maintain soil water around the root tip. So with reference to cohesion and adhesion, obviously questions like this, you want to define cohesion, you want to define adhesion, and then you want to mention and explain how. Okay, so with reference to cohesive and adhesive properties, we have to understand that because water is capable of cohesing, sticking to each other, so water molecules stick to each other, and because the water molecules are capable of sticking to its surroundings, like the soil particles, the water molecules stay close to one another and stay close to one another in a very small space. So that's why there's a high water potential now next to the root tip directly, because they are sticking to the soil. So again, cohesion and then define cohesion. It is the ability water adheres to soil clumps. And then you'd mention here, so yes, water adheres to soil clumps, and so water stays in the clumps around the root tip. Okay, this moves me on to C. Enzymes present in mucilage catalyze the breakdown of organic compounds in the soil. This increases the presence of mineral ions in the soil. State the term used to describe the enzymes that act outside the cell that synthesize them. These are extracellular enzymes. And then he says here, the polysaccharides and amino acids present in the mucilage are the source of nutrients for soil microorganisms that live in the area surrounding the root. And some of these microorganisms can break down soil compounds to release mineral ions. Explain what is meant by a polysaccharide. Okay, so again, let's repeat this again. Polysaccharides and amino acids are present in mucilage, and some of them are active source for microorganisms that live in the area, and some of these microorganisms are going to break down these compounds. And so a polysaccharide, first of all, is a polymer, and like any polymer, it's made of repeating similar or different subunits. In this situation, they're similar subunits, and besides that, they are made specifically of, now in the situation of polysaccharide, they're made of monosaccharides, which are the monomers, and they're bound together by glycosidic bonds. So that's the definition. Okay, so they are polymers made of repeating subunits called monosaccharides, bonded by glycosidic bonds. Now this brings me on to I. So the soil microorganisms use amino acids to synthesize proteins. All the 20 different amino acids that are present in proteins have the same general structure. Draw the general structure of amino acid in the space provided and use this drawing to explain how it's possible to have many different amino acids. So again, amino acids have a same general structure. The only different part about them would be the R group or the variable side chain. So let's go ahead and draw them. So first you start with the alpha carbon, which is the central carbon, connected to a nitrogen and connected to another carbon. The nitrogen here forms an amino group, which is 2 hydrogens connected to a nitrogen, and then the other carbon here attaches to a double bond with an oxygen and then an OH. So this is known as a carboxylic group. And then on top here we're going to have an R group, and then here we're going to have an H. And so this is the alpha carbon. Okay, so this is the amino acid. I don't think they need us to label, but let's just do it to make sure. This is the amino group, and this is the for just be more specific. Yes, so this over here. Okay, what happened here? Okay, so this. Oh, they deleted the drawing. Okay, let's repeat this. So again, an alpha carbon, a double bond. It's okay, you can flip it, it's fine. And then nitrogen with hydrogen. And oh wait, oh yes, and then here we have an R group, and here we have a hydrogen. Now, why it's so different is because the presence of an R group, which is a variable side chain, which is different side chain for each of the 20 amino acids. So for example, glycine has a hydrogen, methionine has a carbon chain with a sulfur. For example, cysteine has an SH, a sulfhydryl group, or a what's it called, SH group, a thiol group. Okay, what else do we have? We also have so yeah, we have different groups, okay, and so each amino acid has a different R group. Now this brings me on to triple I. Minerals are usually present in the soil in very low concentrations. The action of mucilage enzymes and soil microorganisms can help to increase the presence of mineral ions. Root hair cells are specialized for the uptake of these mineral ions and for the absorption of water from the soil. Now, to just explain how presence of mineral ions in the root hair cell can increase the absorption of water by the root hair cells. Okay, that's simple. If you have lots of ions in the root hair cell, you have a low water potential or a really high concentration, and so this low water potential, therefore the presence of the osmotic gradient, allows for the presence of a potential gradient for water, and so water flows down water potential gradient from a region of higher water potential to lower water potential through a partial membrane by osmosis. And so that's it. Okay, let's do this. So firstly, presence of mineral ions decreases water potential inside the root hair cell / increases concentration inside. And so what happens is that water flows, or let's say first creating a water potential gradient so water flows from region of high water potential in the soil to region of low water potential in the root hair cell, and then obviously down a water potential gradient, and they can define osmosis, water Okay, moving on to 3. Cells of the immune system respond to the presence of non-self antigens. Outline the features of non-self antigens. Okay, very simple. So a non-self antigen is going to be an antigen that's present on a pathogen or on a foreign body. Okay, and so the immune system is going to recognize it as being non-self because usually, each cell has its own antigens, and the antigens are, going to be different according to the genes, right? And so in our genes, in your body, you're going to have specific antigens that are, normal and that are self. Whereas for a pathogen, it's going to be foreign because it's different to any other cell in your body. So the difference about, for example, a non-self antigen that's present on a pathogen is that it's detected by the immune system, by, for example, macrophages or by B lymphocytes. And it's a 3-mark question, so I think you need to just write down information. What else do we need to know about non-self antigens? Antigens, you know, they could be made of proteins, they could be made of polysaccharides or oligosaccharides. They could be made of they could be like basically like like glycoproteins. They could be made of glycoproteins, right? For example, for a bacteria, it could be made of glycoproteins. For a virus, it's made of it's not made of capsid. It's found on the capsid. What else can we write about it? I think that's all I can think about for a non-self antigen. It is an antigen found on a pathogen. You can mention examples of pathogens like a virus or a bacterium, or what else? Virus, bacterium, fungus, whatever it is, you know, foreign body. Okay, it could be made of glycoproteins or, you know, proteins in general, depends. And then it is found, for example, for the bacteria, it's found in its capsule. It's recognized by the immune cells like a macrophage, and then which, for example, once swallows the foreign cell by phagocytosis, and then becomes an antigen presenting cell. Okay, this moves me on to B. 4 different types of immune cells are shown in 3.1. Immune system. Okay, now he says complete the table 3.1. Use a tick if the description applies to the name of the cell of the immune system. Use a cross if the description does not apply. Okay, so description of the cell, able to go through a number of cell cycles, clonal expansion. B lymphocytes can definitely go through clonal expansion. Plasma cells cannot. Plasma cells are the cells that are formed from the differentiation of B lymphocytes. So B lymphocytes will undergo clonal expansion, will divide by mitosis, then they will differentiate into the plasma cells, which cannot undergo multiple cycles. And then you have T helper cells, which T helper cells can, and so can T killer cells. He says main role is to secrete cytokines during an immune response. That is the role of the T helper cell, but not any of the rest. What about present during a primary immune response to a virus? In the primary immune response, you're going to have B lymphocytes. Now, plasma cells, are they going to be present in the primary immune response? B lymphocytes are going to produce antibodies when they first recognize the pathogen, but it's going to take a while, but still it's going to be present. T helper cells are present in the primary immune response, and so are T killer cells. Okay, now this brings me on to C. The cell cycle can be divided into different parts. Complete sentences A, B, and C to provide more information about the mitotic cell cycle. The part of the cell cycle that occurs immediately after mitosis. Okay, immediately after mitosis is cytokinesis, where the cell is going to split, okay? And then the part of the cell cycle in which the S phase occurs is interphase. And then he says here, the main event that occurs during the S phase, that's DNA replication. Synthesis phase for DNA synthesis, as well as histone protein synthesis. Now this brings me on to 4. Trypanosoma, something is a unicellular organism that causes infectious disease known as sleeping sickness, insects known as flies, which pass on the organism from infected people to uninfected people when a male and female flies feed on the human blood. So when male and female feed on human blood, just like a mosquito, okay, but it's known as I can't spell this, okay? And you can see that it is a transmissible disease. Now it's in a transmission electron micrograph of the found in human blood. So it's transmitted by a vector just like malaria. And he says here, draw an arrow in figure 4.1 to indicate the location between the cell where ribosomal RNA and proteins are assembled to make ribosomal subunits. So to make ribosomal subunits, that's going to be in the nucleolus. And so the location here, this is the arrow. I'll draw the arrow to the nucleolus. and I'll label it, okay, nucleolus. Okay, this moves me on to, with the reference to figure 4.1, explain how the structure labeled X provides evidence that T brosi is motile and able to move. Okay, let's see. So evidence it's able to move, they have a labeling here on X, and you can see actually there's an axonemal structure here, which is similar to the structure of flagella or to the structure of, well, obviously eukaryotic cilia. And so you can see here that there are two, two singlets of microtubules, and then 9 microtubule doublets. And so this is known as the 9+2 structure, which is a structure found in flagellum, and it's found, wrapped with cell membrane. And besides that, so yeah, basically this is important for movement because this is highly motile, because there's movement with dynein protein arms and everything. So explain is that there is the microtubules in an axonemal structure, and this axonemal structure is a 9+2 structure, just like flagellum. So I can assume that this is flagellum, so to move it. And yeah, so I think that's all I can say. It's only 2 more questions. I think axoneme and flagellum cover the mark. Now this brings me on to 3. With reference to figure 4.1, explain whether T is a eukaryote or prokaryote. It's obviously a eukaryote. There's a nucleus. There is, obviously structure X. You can see how there is an axonemal structure in the flagellum, whereas for flagellum and maybe like a bacteria cell, it's actually going to be, first of all, outside the cell. Second of all, it's not going to be made of microtubules. It's going to be made of a series of identical proteins, and it's going to move in a very specific corkscrew-shaped movement. But other than that, you also have, membrane-bound organelles. You have a mitochondria. You have what else do you have? Yeah, so the nucleus itself, it covers it on its own. So let's go ahead and write that down. Okay. Eukaryotic. So a nucleus, a nucleolus. What else? It has linear DNA, or you can say chromatin. You can ask me, how do I know it has chromatin? It's very simple. We look at the nucleus, see those dark areas over here. These are actually all heterochromatin, right? And there's like euchromatin here, but it's very light, and that's why it's like dark here, but like again, not so dark. So yeah, it has chromatin, linear DNA. What else does it have? It has histone-bound DNA, obviously, but this is not shown. With reference to figure 4.1 specifically, it has mitochondria, which is a membrane-bound organelle. And lastly, I could talk about the flagellum here. Okay, now this moves me on to B. Malaria is an infectious disease caused by organisms belonging to the genus Plasmodium. State the term used to describe an organism that causes disease. So the organism that causes disease is the pathogen, but I think that here they're talking specifically about the organism. Yeah, so it's a pathogen. Disease caused by organism belonging to the genus Plasmodium. So state the term used to describe an organism that causes disease. It's a pathogen. Okay. Name one type of species of Plasmodium that causes malaria. So there's Vivax, Plasmodium vivax, Plasmodium ovale, Plasmodium falciparum, Plasmodium malaria. So you could just write any one of them. Now this brings me on to outline the similarities and differences between the models of transmission of malaria and sleeping sickness. So again, they're both transmitted by a vector, they're both transmitted by an animal, which is a fly. Now in the case of malaria, it's transmitted by the female Anopheles mosquito. Here it's transmitted by both male and female flies, which, yeah, both of them are going to feed on human blood. Both of them are going to carry, like for malaria and for this disease, they're going to carry the, pathogen within their body, and it's going to undergo certain type of, life cycle parts in the mosquito's body. So the mosquito acts as a vector, and it transmits the pathogen then into the body of the uninfected person. So I think that is like similarities and differences. So he needs similarities and differences between the host. So both which is a fly. Both are going to be by the fly. However, malaria, okay, it is since only the female, yeah, it takes blood drinks, really. Okay, now this brings me on to 5. Nucleotide and nucleoside analogs are therapeutic drugs that have a similar structure to nucleotides or nucleosides of RNA and DNA. A nucleoside is composed of an adenine base and a pentose sugar just without the phosphate group. And he's saying here the names of the bases present in RNA and DNA nucleotides can be abbreviated using a single letter, and these are shown in figure 5.1. So here you see the abbreviations of the bases A, C, G, T, U. Now complete table by naming the bases and writing whether it's a purine or pyrimidine, and identifying whether the base is present only in RNA, write RNA in the table, only in DNA, write DNA in the table, or in the RNA and DNA, write both in the table. Okay, so name of base for A, that's adenine, for C, that's cytosine, G, that's guanine, T, that's thymine, and U, that's uracil. For purine or pyrimidine, adenine and guanine are purines, so purine, whereas all the rest are pyrimidines, so cytosine, pyrimidine, thymine, pyrimidine, uracil, also pyrimidine. Now, present in DNA or RNA, adenine, guanine, cytosine are present in both, but thymine is only present in DNA, and uracil is only present in RNA. This brings me on to B. Okay, so it's an analog drug used in the treatment of some viral diseases. It enters a cell infected by the virus and is metabolized by the analog, carbovyryl triphosphate. So basically it is metabolized to this guy. So this is over here an analog drug, which is basically a nucleotide analog. And then it's going to be metabolized by the virus into this analog. Okay, so again, it's a drug and it's going to be entering the cell infected by a virus, and it's metabolized to this guy, and so shows the molecular structure of this guy and the carbovyr. Okay. Now this brings me on to carbovyl triphosphate. It can be inserted into the elongating polynucleotide chain instead of a nucleotide. This interferes with the action of DNA polymerase during the synthesis of viral DNA, and so stops the formation of viral DNA. So again, this is very important. I think he's talking about the mode of action of this drug. So first, when it enters the cell metabolizes it from this nucleotide into carbovyr. Carbovyr then is going to be added onto the elongating DNA chain that is formed by DNA polymerase, because this chain is going to be now synthesizing viral DNA, and this viral DNA is going to now control the cellular machinery and is going to control the cell and cause it to be an infected cell. So what happens is that this nucleotide is going to insert, and when it inserts, this is going to stop the DNA polymerase from working, because it could be that maybe it cannot attach to other nucleotides. B, it could be that the DNA polymerase, obviously we know that it does a proofreading mechanism where it stops when there's something added that's wrong. And so DNA polymerase identifies, oh, this is wrong, or maybe it does not fit, for example, into the substrate binding site of DNA polymerase, and so it stops synthesizing the viral DNA. And so with reference to figure 5.1, explain whether carbonyl triphosphate would replace a purine or pyrimidine nucleotide in an elongating polynucleotide chain. So is it purine or pyrimidine? Actually, it's purine. So it could replace adenine or guanine, because you can see it's a double ring structure. So whenever I want to identify is it purine, it's pyrimidine, I look at the ring structure. If it's a single ring that's made of 6 elements, okay, that's going to be pyrimidine. If it's a double ring, one made of six elements and one made of 5, that's going to be a purine. So it is a purine because it has double ring structure. This brings me on to double I with reference to figure 5.1 and the action of DNA polymerase suggests why the conversion of this guy to carbovir increases the chance of analog being added to the viral polynucleotide chain. Okay, let's see. So why is the nucleotide analog, okay, the one that is added, why is the carbovir the one added instead of abacavir? So the difference between the two is that abacavir is like a nucleoside, okay, because it only is made of this sugar and then a base, whereas here you can see there is a phosphate group, a phosphate group, and so the phosphate group is the reason that it's added, is because we know that DNA polymerase only adds phosphorylated nucleotides, and so that's the reason, okay, because again the carbovir triphosphate is phosphorylated, and so since it's phosphorylated and DNA polymerase only adds phosphorylated nucleotides to the growing chain. This moves me on to triple I, suggesting explain how carbovir triphosphate interferes with the action of DNA polymerase and how this may prevent the synthesis of viral DNA. Well, again, I think we've already explained that up again. So carbovir triphosphate, when it's going to be added, what happens is that maybe DNA polymerase does not have it, it does not fit, for example, into the DNA polymerase active site. Maybe, it cannot keep on working. Maybe there's a proofreading mechanism. Maybe this carbovir triphosphate acts as an inhibitor to the enzyme, so basically works as a competitive, non-competitive inhibitor. There could be a chance of that. It could be that, once you add this nucleotide, again, because of proofreading mechanism, we stop the synthesis of viral DNA. So these are all different suggestions, right? So just anything that comes off your mind, and then make sure to explain it so much that you can guarantee a grade. Okay, so let's do this, okay? I think, yes, maybe carbovir could act as an inhibitor to DNA polymerase. Maybe it has maybe an affinity I think that it should be a competitive inhibitor. Maybe it's a competitive inhibitor. Actually, I don't think it can work as an inhibitor because it's already attached. The idea that the inhibitor is that it does not allow the... Let's just write it down as a suggestion. So competitive inhibitor, since it has similar structure to purine nucleotides. Maybe DNA polymerase has a proofreading mechanism and DNA chain breaks down. What else do we have? We also said that this idea that it cannot form bonds. Overall, you know, since okay, is to catalyze, you know, the formation of phosphodiester bonds. So if that does not happen, then you basically interfered with that function. And these are all my suggestions. Feel free to add more. Now this moves me on to 6. Okay, so the Weibull-Long model was developed after an extensive study of the gas exchange system, and the model includes detailed measurements of the airway diameters, that is, lumen diameters of the airways in the model, and the different parts of the gas exchange system are identified with the generation number as outlined in figure 6.1. So you have a generation number, and you have the airway. So first we have the trachea, then we have the main bronchi, left and right, then we have the bronchioles. Actually, we have secondary bronchi. Okay, so here we have more bronchi, and then here we have bronchioles. Once you reach into bronchioles, you have different kinds. You have terminal bronchioles right here, and then you have respiratory bronchioles, which have a similar function to alveolar ducts, which connect you to the alveolar sacs or the alveoli. Now this moves me on to, yes, this is the generation number. What does generation number mean? I'm not really sure. So it says Weibull-Long model describes how each airway divides into 2, and he says name the long airways in generation 1, labeled X, in figure 6.1, the branch from generation 0. So generation 0, what branches here? Generation 1 is going to be the right and left bronchi. And this brings me on to B. The airways are well supplied with blood, however, the airways from generation 0 to generation 16, shown in figure 6.1, are not able to carry out gas exchange. Okay, so the airways are well supplied, but the airways from generation 0 to generation 16 cannot do gas exchange. All they do is they conduct the air so that it reaches to the terminal respiratory bronchioles and alveoli, where respiration is going to happen. Now, suggest why the airways from generation 0 to 16 are not able to carry out gas exchange. It's very simple, okay? To carry out gas exchange, you need to have structures like those of the alveoli. You need to be very close to blood capillaries so that they can exchange gas with something. You need to be having a very great surface area, which they clearly don't, okay? And you need to have, besides the surface area, you need to have a thin diameter, thin section, or thin, what's it called, wall, so that gas can easily move in and out, which, for example, for trachea, that is covered by cartilage, or for bronchi, that's also covered by cartilage and with smooth muscle and everything, that's very, very impossible. So that's the reason, okay? Again, here you can write down, so no close, blood capillaries, very small surface area, very thick sections, so gas, it can't be exchanged, so it can't be exchanged, it can't diffuse into and out of these structures. Now C, high resolution computed tomography, HRCT, is an imaging technique that is used to obtain measurements of airway diameters in people with pulmonary lung disease. Regular repeating imaging must be avoided because it involves exposure to harmful radiation. Scientists research the use of different techniques, that is basically HP gas MRI, that does not involve harmful radiation, and 2 types of MRI imaging known as projection and multi-slice were used to obtain measurements of airway diameters. Now figure 6.2 shows the results of HP gas MRI study compared with Weibull-Long model, the 4 generations 0 through 5, and the Weibull-Long model was used as a standard reference for the airway diameters. Okay, so the airway diameters, we have projection, multi-slice, and Weibull-Long model. Okay, so you can see this is the standard okay, diameter. And then here we have the comparisons with multi-slice and with the projection. He's saying here, state the relationship shown between the area diameter and the area generation from the Weibull-Long model. Okay, so from the Weibull-Long model in general, as you increase in generation, as you increase the generation number, you can clearly see that the diameter decreases. Okay, I don't know why I keep highlighting the wrong one. Yes, so you can see the diameter keeps on decreasing until reaching the generation 5. So there is a relationship which is basically as airway diameter as airway generation increases, diameter decreases. Okay, with reference to data in figure 6.2, explain whether HP gas MRI could be useful alternative to HRCT in obtaining airway diameter measurements. Okay, so it could be useful in obtaining airway diameter measurements. So he specifically is asking about which one? About HP gas and MRI. Okay, MRI could be useful alternatives to HRCT in obtaining airway diameter measurements. Now, you can see the difference between the two types of MRI known as projection and multi-slice. And yeah, so basically HRP gas MRI in general, they are different in results. However, I think that they're very close in diameter. Like the difference is not that huge. You know, here it's for example 1.8. The difference for example for the projection and for this guy, it's much less. However, I think that each time it's different. Okay, I think they could be useful alternatives. The difference is not that big. However, maybe like try to use the average between the two, or like I think that we could try to use the average between the two. Okay, and so the reference to the data, could it be useful? Okay, we need to reference the data here. So as I mentioned here, we have 1.4, 1.6, that's a 0.2 difference, 0.2 over 5. That is like, I think it's like 0.04. So it's 1.44, 1.48, 1.52, 1.56, and then 1.6. So at this point over here, you have like 1.52. And then here you have it at 1.8. And here you have it at 1.64. So the difference is very small. And it keeps on decreasing actually. The difference is not that big, for example, here. Right? So I think that, just make sure to write down some of these measurements. So, you can see here that the difference is not that big, and I think that since it's a 3-mark question, you can just use one data plot. But the whole idea is that, in general, as you can see, when you increase the airway generation, you are decreasing the diameter, and that's the most important part. You're not using harmful radiation, and so I think it is useful. So let's write that down. Okay. Here you can mention an example. For example, for the multi-slice, it is like only 1.64. For HRCT. Okay, but is this the first generation or zero generation? Zero generation. Yes, 1.8 centimeters for HRCT, and overall okay. And lastly, you can see here that what else did we say? Oh yeah, it's not using harmful radiation. And okay, I think that is all. So, and you can also maybe write, if you know this, that computer tomography is actually much harder to do than, for example, MRI. The MRI machine is much simpler, and it's just like easy for the person to go in and come out. But like HRCT, it's much more scary. Like generally looking, it's much more scary, and it's much bigger, And so yeah, generally people prefer with the MRI over the CT, especially with the radiation. Now that is all for this question. Okay, so that's all for this video. I hope this video was useful for you guys. If it was, make sure to like, share, and subscribe, and I'm gonna see you guys in my next one.